I I am going to be, instead of sending it from the Tree Steward training account, I'm going to send it from my personal account so that because the way it's going to work is you're going to get an email from me with a couple of little quiz questions in there. Just hit reply and write your answers in to the reply. I'm going to blind copy everybody so that we don't all get bombarded with emails and stuff and we don't see each other's answers. So anyway, expect that later tonight. And if you don't get a quiz, send an email to the Tree Steward training address and we'll figure out what's going on. But anyway, look for that later tonight. And the questions will be easy. So and it's open book and it's not graded. Just gonna review it and just make sure that people, kind of got the basic concepts. So anyhow, that's it.
All righty.
And I'm Noreen Hannigan, by the way. I wasn't able to make it to the welcome class, but so it's my first time getting to meet people. Okay, I'll turn it over to Laurie.
All righty. Well, I'm going to introduce Jim. Jim's our speaker, our teacher, instructor extraordinaire for this evening. Jim McGlone has a PhD in human ecology, also known as economics, from Virginia Tech, and a master's certificate in environmental law and policy from the U.S. Department of Agriculture Graduate School. A self-taught naturalist and ecologist, he has managed the natural resources of a 500-acre park in Fairfax County and is a retired urban forest conservationist with the Virginia Department of Forestry in Northern Virginia. He continues to teach environmental stewardship through TreeStewards, master naturalists, master gardeners, and other groups. He and his wife have been practicing conservation landscaping since 2005 and removed turf grass from their property in 2008. He is a certified arborist and has a tree risk assessment qualification, both through the International Society of Arboriculture. All right, take it away, Jim.
Okay. So, welcome. Thanks for joining us tonight. We are going to be I'm going to be spending the first half on tree anatomy and physiology. If you have questions, you can put them in the chat box and I will periodically stop and answer your questions. So with that, let me just organize my screens a little bit better. There we go. And so let's get started. So if we're going to talk about tree anatomy and physiology, probably the first question we need to ask is, what is a tree? And it turns out that the Linnaean system of binomial taxonomy really doesn't help us out. This is a system, you may be familiar with it. You start with plants, and the first step is you divide them into vascular and non-vascular plants. Then you divide them into seed-bearing and spore-bearing vascular plants, and then divide the seed-bearing into what are called the naked seeds, or the gymnosperms, and the covered seeds, which are the angiosperms, which further get divided into the dicotyledons and the monocotyledons, or as they're more commonly called, dicots and monocots. And you can see these are the examples, and this actually follows the evolution of plants pretty closely. The first terrestrial plants were non-vascular plants like our modern mosses, and then they developed vascularity, and we got ferns and horsetails, and then conifers and ginkgos around here anyway would be the gymnosperms. And most of the angiosperm trees around here are dicots, oaks and maples. The monocot that everybody agrees is a tree is a palm, but bamboo also is a woody monocot. And the problem is that all of these lineages have tree forms in them. So trees evolved separately several times over the history or the evolution of plants on land. In fact, the first trees were just bundles of vascular tissue. the trunks were. But they got to be, some of those fern trees got to be 200 or 300 feet tall. So they were big things. So how do we deal with this? Well, a tree is a plant with a perennial woody stem. That's the simplest way to differentiate them in plants. When we start talking about trees and shrubs or trees and herbaceous plants, it's really, we divide them into woody plants and non-woody plants. And the woody plants have that perennial stem. The non-woody plants, the herbaceous plants, they all die back to the ground every year and then resprout and grow new shoots from the ground. Whereas the woody plants maintain that perennial stem and they grow new shoots on top of that perennial stem. If you look at your tree steward manual, they will define a shrub as having the potential to grow to at least 15 feet tall. Other definitions of shrubs start at 20 feet, others 25 feet. They usually make some statement about single trunk that may divide into 2 or more trunks or multiple trunks. And they all make the point that they do stand by themselves. So it doesn't matter whether we're talking about a tree or a shrub. If it's a woody plant, it's a self-supporting plant. Sibley, in his fairly good book on trees, in the introduction, distinguishes between a tree and a shrub by saying you can walk under a tree and you walk around a shrub. However, if you've ever seen a open grown holly or southern magnolia or eastern white pine, you know there are some plants that everybody agrees are trees, but you still have to walk around them. And in fact, those plants do occur in his book. From my point of view, I really don't make much of a distinction. I don't make any distinction between trees, shrubs, and woody vines, at least as far as their anatomy and physiology is concerned. They all work pretty much the same way. There are some structural differences due to the size of the plant. Obviously, you don't need the same kind of a trunk to support a canopy that has a 10-foot radius as opposed to a canopy that has a 50-foot radius. But otherwise, they all act the same. So everything we talk about tonight is going to apply to any woody plant. So let's look at the structure of trees. First of all, this is what trees aspire to. They don't all make it to this, but this is what they want to look like. This is how they want to grow. Contrary to what a lot of people believe, the root system, aside from being dendritic, that means it splits into smaller and smaller pieces the same way the canopy does, looks nothing like the canopy. However, and this is important, especially when you get to tree planting, the mass of the canopy and the mass of the root system are more or less the same. So there's a high degree of correlation. But as you can see from this picture, the geometry is very different. One of the things that occurred to me when I was trying to explain this to people is think about making pizza from scratch. So you mix your water and flour and whatever else you put into pizza dough and you mix it up and you leaven it and do all that stuff. And then when you're getting ready to make the pizza, you start off with a ball of dough, which you then compress and stretch into a disc of dough. So that ball of dough is like the canopy of the tree. The disc is like the root system. And the reason the root system is in the top of the soil column is because roots not only provide support and gather water and nutrients, they also gather oxygen. And they can find water and nutrients deeper in the soil, but they're not going to find enough oxygen deep in the soil. That's why they get spread out in the top 24 or so inches of soil. In terms of functionality, the roots and the branches of the canopy are similar, but they're not quite the same. So in the root system, we have fine absorbing roots. These are small roots, small structures that support the energy system through collecting water. The in the branches, we have leaves. These are the small structures that support the energy system through the collection of light and carbon dioxide. As we go from those fine absorbing roots towards the trunk of the tree, we get conductive roots, which are larger. They do still do some absorbing, but a big part of their function is to collect and support those absorbing roots and move the water that is collected through the system to help support the energy system. The similar structure would be the branches and twigs, which are the larger stems in the canopy that collect and support the leaves. Then right at the base of the trunk, right at the base of the tree, you have the buttress roots. These are very large roots that connect the root system to the trunk of the tree, but they also are primarily responsible for supporting the tree. Similarly, in the canopy, you have scaffold branches, which are the large branches that connect to the crown, and the branches and twigs connect to them, and then the leaves. So they are the last conduit till you reach the trunk. And the trunk kind of ties everything together. It supports the canopy. It is connected to the root system. And this is where water and sugars are flowing. It's the main thing that's going on in the trunk of the tree. There are three primary types of tissues in trees. The first type is meristem. These are cells that are capable of dividing and making new additional cells and differentiating into the other types of tissues. And all trees have two types of meristem. They have primary meristem, which is what makes the shoots and the roots. This is where elongation takes place. And there are apical, lateral, and axillary buds that make up this primary meristem. The apical buds are at the ends of the tips of the roots and the branches. The axillary buds are typically they're found in only in the canopy, and they form in the axil between the leaf and the twig. And then the lateral buds are scattered throughout the tree. The secondary meristem is what makes the new wood. This is where diameter growth occurs. It's also called the cambium. It's found right below the outer bark of the tree or the bark of the tree. And in some trees, the ones that have thick bark, there is also a thing called a cork cambium. And this is what makes cork in the outer bark. So not all trees have cork cambium. The thin bark trees, things like beech and maple and holly and American hornbeam, do not have a cork cambium. The next type of tissue is called the vascular tissue. And in angiosperms, we call this vascular tissue vessels. And in gymnosperms, they're called tracheids. Vessels are an evolutionary form of tracheids. We believe that they evolved from tracheids. They are both similar in that they are long tubular cells that are dead at maturity, and they conduct the water and nutrients from the roots of the tree up to the crown. Now, some people call this xylem, which is Fine if you're talking about herbaceous plants, because they don't generally have much else in their stems besides xylem. But xylem is the Greek word for wood. And in the angiosperms, especially the dicots, wood contains vascular tissue, fibers, and parenchyma. In the conifers, the wood only contains vascular tissues and parenchyma because they don't have fibers. I will use the term xylem to refer to the wood, not just the vascular tissue found within the wood. And this will be clear in the next slide. There is also phloem. This translocates photosynthate. That's a fancy way of saying it moves sugar around the plant. The third type of tissue is called ground, and it's everything else. So in plants that have fibers, fibers would be part of the ground. In angiosperms and particularly dicots, fibers are found in the stems and the seeds. These are dead cells that give structure and support to the plant. There are epidermal cells. There's also epidermal or the epidermis. This is the outermost skin of the plant. It includes, but is not limited to, the bark. And the main thing that happens in the epidermal tissue is the control of gas and water exchange. And depending on which camp you are in, the epidermal is either also dead cells, or it could be, could have some live cells. And that's because of the final type of ground tissue, which is called parenchyma. And this does lots of things. This is where photosynthesis takes place. This is where the tree stores carbon, either by switching sugars to starches, or switching starches back to sugars. This is where, when the tree is wounded, the parenchyma is that first line of defense. It kind of causes a scabbing over the wound. You're going to do pruning on the weekend, and this becomes important. So when you make that pruning cut, immediately the parenchyma starts to create a scab over that cut. It's not something that you can see, but it's there as part of the defense of the tree. And the parenchyma is also where plants make their or create their secondary chemicals. These are the things that plants create to defend themselves primarily from pathogens, but also from wood decay organisms and insects. So the parenchyma, which is found in lots of different places, are living non-reproductive cells. And some people classify the guard cells in the leaf stomata, which are the pores. That's what causes them to open and close. Some people say that's parenchyma. Other people say it's an epidermal. That's why there's that differentiation to whether or not epidermal cells are alive. So we can look at a cross section. And if we start, we'll start at the outside. Let me try and be fancy here. We'll start at the outside here, and you have the outer bark, which goes into this area. And you can see there are some big cells in here because this is a Quercus nigra, which is an oak, and it has cork cells in it. So underneath these big cells, I've drawn this red line, which is about where the cork cambium is. So this is living tissue that can divide and make these big cork cells. And then between the cambium, which would go in this trench here, And the cork cambium is where we find the phloem, which is this kind of finer grain stuff. And this is the net that moves sugars around in the plant. When we go inside the meristem or the cambium, we find the wood or the xylem. And within the xylem of these dicots, you have vessel elements. These are these big openings that you can see. And also, not as obvious, but in these lines through here are also vessel elements. They are surrounded by parenchyma, which helps these vessel elements in their functioning of moving water up and down. We also see rays of parenchyma throughout the xylem of the tree, and notice that these rays go all the way out into and connect with the phloem. So that's how the sugars get into the parenchyma, where it gets stored or is burned to do whatever the parenchyma needs to do. And then finally, in dicots or angiosperms, you also have fibers. And this is what gives a lot of strength to the wood. That's this kind of khaki or tan colored stuff. And the cream colored stuff is parenchyma. So that's what the stem of the tree looks like and how the parenchyma or the meristem there works. When we look at a twig, you have buds. So this is an apical or terminal bud up here. These are axillary buds. One of the ways you identify oaks in the winter is they have a cluster of buds. This happens to be a northern red oak. These are axillary buds. And you can see below some of these, the leaf scar where there were leaves last year. This one's missing one of its axillary buds. And then down here is a terminal bud scar. If you can find the terminal bud scar, then the distance between the terminal bud and that terminal bud scar is how much the tree grew in a single year. The other thing that's not labeled in here, these little white dots, are lenticels. So that is part of the epidermis, and that's where gas exchange is taking place. But in this case, it's oxygen going in and carbon dioxide going out. And we'll talk about that in a minute. The thing you don't see in here are lateral buds. These are suppressed buds that are left behind as the twig grows. Okay. So far, no questions. Or are there questions? All right, then we're going to move on. So tree physiology. This I consider more of the way trees interact with their, or not interact with their environment, but the way trees control themselves, and in particular, the energy system of the tree. So plants create biochemical energy, which is sugars, through photosynthesis. And they take sugar, or sugar, water, and nutrients get moved around the plant through the vascular system. And in living cells, that sugar is burned in the presence of oxygen through cellular respiration. This is the same thing that happens in our body. It's also known as the Krebs cycle, and this energizes the adenotriphosphate. And ATP fuels the production of all the biochemicals biochemicals from the nutrients that are taken up through the roots and are used to repair the cell, to create those secondary chemicals to defend the plant. And if it's meristem, could be making new cells or new tissues. So we see photosynthesis taking place in the parenchyma, respiration taking place in the parenchyma and the meristem. And together, this process of photosynthesis and cellular respiration are turning solar energy into cellular energy. So if we look at photosynthesis, which takes place in the green parts of the plant, and you can see in this picture, these Palisade parenchyma are the cells that have chloroplasts in them. So these green dots are the chloroplasts where photosynthesis takes place. And the basic chemical equation for photosynthesis is 6 carbon dioxide molecules and 6 water molecules get rearranged using solar energy into a single glucose molecule and 6 oxygen molecules. And the carbon dioxide comes from the air through the stomata. The water comes from the roots through the xylem. You got sunlight out there, and the output of this process is sugar with a waste product of oxygen. And so this also shows you the cross section of the leaves. Here's the vascular tissue. This connects up with the rest of the tree. Here are those guard cells that some people regard as parenchyma and others regard as epidermis. These are definitely living cells because they change shape to open and close this pore to let gases into the interior of the leaf. Once the plant has the sugar, it undertakes cellular respiration. And cellular respiration is this equation, and notice it's a glucose molecule plus 6 oxygen molecules that leads to 6 carbon dioxide molecules, 6 water molecules, and 3 energized adrenotriphosphate molecules. And the input in this process is sugar from the leaves, oxygen from the lenticels, and most of the tree's lenticels are in the root system. So that's why they need to be in the top 24 inches so they can collect that out, the oxygen. And the output of this is ATP that goes off to the ribosomes, where the ribosomes use the ATP and nutrients to make new tissues and toxins and for cell maintenance. And the waste product of this is carbon dioxide and water. So what the tree is doing is, through these chemical processes, is it's just cycling carbon dioxide and water to create energy that the tree can use for its life functions. And this all leads to a model that I put together. And you'll see this again when I come back to talk to you about tree diseases. But you have your leaves up here that are doing photosynthesis, your roots down here, water is going up to the leaves, sugar is coming from the leaves down into the root system for storage, but it's also going to those living cells where they're also using nutrients and oxygen from other places to create the new tissues, to maintain existing tissues and create those toxins. So by looking at what is happening or not happening, and using this model, it can help you zero in on a pathogen or a pest within the tree that is affecting the life cycle of the tree. Now, trees don't just use energy for their own purposes. They also interact not only with the soil ecosystem, but with the above ground ecosystem by exuding trees or exuding sugars to attract microbes. And it says here beneficial, but it can also be those microbes that really are neither beneficial nor pathogenic. They're just there, and they're trying to attract those microbes in part for defense. So by coating their exterior surfaces with non-pathogenic bacteria and fungi, that creates an additional layer that the pathogens have to get through in order to attack the tree. And many of these microbes are beneficial because there is a whole lot of warfare that's going on in the soils and otherwise where bacteria are attacking other bacteria, which is where we get most of our antibiotics is from soil bacteria. But you also have bacteria attacking fungi and fungi attacking fungi and fungi attacking other or bacteria. And there are even some fungi and bacteria out there that will be pathogens or will attack larger organisms, particularly nematodes, which can attack tree roots and create problems. The other thing the trees are doing is attracting mycorrhizal fungi. So mycorrhizae, if you just say this word, that means fungal root. And so you have to have the fungus and the root to make the mycorrhizae. That's what all this fluffy cotton stuff is on the root of this plant. What the fungus is doing is it's reaching out with its hyphae and collecting water and nutrients and exchanging that for sugar. Some of these mycorrhizal fungi specialize at the family level or above. And there are many of them that really don't care what plant they're hooking up with. The plants are also not overly picky about what mycorrhizae they will work with, mycorrhizal fungi they work with. Some oaks have been recorded as having more than 20 different mycorrhizae because they're working with 20 different species of mycorrhizal fungi. The other thing is the spores for the mycorrhizal fungi are everywhere. So I've heard people talk about, well, we don't have the right mycorrhizal fungi for this plant in this area, and that doesn't really hold up to scrutiny. And you can also buy those spores at the nursery to inoculate your soils, but there is no science that suggests that that does anything. In fact, what the science points to is that's like taking a pail of sand to the beach. It just doesn't make any difference. And now I do have some questions. Let's see. Nora signed me up. How do you know it's a year's growth? Well, because, and that refers to, let me get back here. Oops. And get this out of the way. So at the end of the year, or when this bud forms, you can see these little scaly plates on them. And that makes a scar where those plates attach to the twig. And this is where the terminal bud was last year. And so when the terminal bud broke and it started making a new shoot, it kept going until it made the terminal bud at the end of this growing season. That's how we know it's a year's growth. If it was more than a year's growth, then there would be another terminal bud scar that goes all the way around the twig somewhere in between. And if you can find more than one terminal bud scar on the twig, that's great, because then you can see how well the twig did in one growing season versus another. There is absolute, Jackie asks, what distinguishes a tree with a cork cambium from a cork tree? As far as I know, there's only one cork tree, which is an oak tree, and it has a cork cambium because that's where the cork comes from. Trees that do not have a cork cambium do not have cork in their bark. And Pete is correcting me on ATP. They may have changed this designation since I took Oh, you're not correcting me, you're just taking notes. Okay. I learned about the Krebs cycle back in 1975 when I was in high school, so things may have changed since then. And I guess that's it. Okay. Problem is I keep going back and forth between two different, all right, tree growth. How does a tree grow? Well, remember, trees only add new cells in their meristems. So we have the primary meristem, which are the buds, and the secondary meristem, which is the cambium. And the primary meristem is responsible for elongation of the tree. This is what makes the tree taller. This is what makes the branches and the roots longer. And as those buds wake up and start producing new cells, they produce the cells, the cortex, which becomes eventually the vascular tissue of the tree, the epidermis. They're going to be producing any lateral buds or axial buds or leaving behind any pieces of primary meristem. Some of it will differentiate to become a leaf. Some of it differentiates to become a flower. But all of this stuff that's associated with elongation and spring growth is in the primary meristem. The secondary meristem, as I pointed out already, is located between the bark and the wood, and it produces the xylem, which persists as wood, and this is where water and nutrients are moved up from the roots to the canopy. That's the primary function. Although in late winter, xylem can also be moving sap and photosynthate that's been stored as starches over the winter. That's why we have maple syrup. And then phloem, which may become the bark or be reabsorbed into the tree. It depends on the species how they do that. And this is where we move sugars from what we call a source to a sink. So right now, sugars, the source of sugars in the tree are the leaves. The sink is going to be any living tissues that need sugar for cellular respiration, but also any of the parenchyma cells where it's going to get converted into starch and stored over the winter. Starting generally in December, very late December, the source can be those xylem cell, or not xylem, parenchyma cells that are storing the starches and converting it back to sugar, and then it gets moved up into the canopy to support the spring growth. So this is what it looks like. So the blue part is a twig from last year that has a terminal bud here. The terminal bud wakes up and it produces a new shoot that may have some lateral buds or a new terminal bud. Once the primary meristem gets going, then the secondary meristem kicks in and we see diameter growth with a new layer of wood on top of last year's growth. And the cool thing is, it doesn't matter whether this was a germinated seed from last year that made this twig, or whether this is a twig at the top of a 250-foot tall coast redwood. The same thing is happening. Trees are modular that way. So this is what you can see up here where you have brown bark. This is last year's epidermis that is getting toughened up and turned into bark. There was a terminal bud there, and this was a pretty good growing year because this viburnum dentatum elongated quite a bit. And as I was preparing this, I realized, what's happening from here on in a tree is the same thing that happens in an herbaceous plant, except that with the herbaceous plant, the stem always dies back to the ground. And so the primary meristem up here that makes the shoot in an herbaceous plant is always located at ground level or at the top of the root system. Whereas in trees, it's located at the end of last year's growth. So in some sense, trees are growing new plants on top of the old plants. So that's the elongation. The other part of tree growth is diameter growth. So what happens every year is you've got the end of last year's growth. So you've got a twig from last year, and then you get new wood, new xylem growing on top of it. Now, keep in mind, at this point, all of the xylem is what we call living tissue or living xylem because it has living parenchyma in it. Eventually, what will happen, if the tree lives long enough, depending on the species, it will get to the point where it starts to abandon its older xylem, and it's no longer using this part of the tree to move water. And there are no living cells in here. The parenchyma, and I'll talk about exactly what's happening here in a minute, but the parenchyma in here has died, and in that process has created what we call heartwood. That's Heart, as in the core of the tree. And this is dead xylem as opposed to the living xylem that still has living parenchyma in it. This is what gives us the growth rings of a tree. So here's my water oak again, and the two red lines measure a year's growth. So this is where it stopped growing last year. Then it grew these great big vessel elements, and then it grew a lot of fiber and smaller vessel elements, and finally shut down its growth here. So between this red line and the blue line is what we sometimes call early wood or spring wood. And then from here, from the blue line to the next red line is the late wood or summer wood. And both of these things, early wood and summer wood, make a single year's growth. Water oak and the oaks in general, it's pretty easy to count the tree rings because they're what we call ring porous. That means it produces early in the year these great big vessel elements that can move a lot of water, and then produces more wood fiber and more and smaller vessel elements through the rest of the year. Sugar maple, on the other hand, and other trees of its this kind are this called we call ring diffuse, and what that means is the vessel elements that it produces over the course of the year, as you can see from this slide, are about the same size, whether it's produced early in the year or late in the year. And in the case of these ring diffuse trees, it can be really hard to count the rings, and you really don't have that early wood, late wood that you get in the more ring porous. And of course, there is a continuum between those oaks, which are very definitely ring porous, and trees like sugar maple, which are very definitely ring diffuse. And it goes by partial ring porous, or they've got a lot of different names in it. But just to help you understand, these bright lines are the parenchymal rays that we talked about before. This is where the growth rings would be. So you can see it can be very hard in some species to count the growth rings. Heartwood, as I said, is these are dead tissues. It's created by the parenchyma as they die. What the parenchyma does is it creates a lot of secondary chemicals and infuses them into the wood, into the fibers. In some species, The parenchyma will blow tyloses or airbags into the vascular, into the vessel elements to clog those up. Sometimes they're putting sap in those things. The tree stops moving photosynthate into the heartwood because it's no longer needed there. It's dead tissue. And usually the heartwood is darker than the sapwood. You can see this. I think this is a poplar, a tulip poplar, Liriodendron tulipifera, but you can see the sapwood. So there's living parenchyma cells in there and then dead parenchyma cells in here. One of the big distinctions between heartwood and sapwood is sapwood uses active defense if it gets attacked by a pathogen or a borer or a grazer or whatever, because it has the living cells that can create those defensive chemicals in response to the attack. The heartwood only uses static defenses. It cannot change itself in any way. It simply has to resist the rot with the resources that it has. The other thing that we get from in terms of growth, particularly related to diameter growth, is reaction wood. So if you look at this tree, you can see something bent it, and then it started growing upright again, but you can see this ridge of wood that it grew in response to that bend. And what's happening there is the cambium, the secondary meristem, when it's compressed or stretched, will respond to that physical stress by growing more wood fiber. So when you look at the cross section of the tree, you can see there's the heartwood of this branch. And then you can see that it grew a lot more wood fiber, a lot more wood on the underside that was under constant compression than it did on the top side, which was under constant tension. The other place you'll see this is when trees sway in the breeze, they will build what is called a taper. And that's because as the tree sways in the breeze, the cambium is being put into tension and compression alternately, and it responds to that by creating more wood to strengthen that bending point. So if we look at, and this is kind of a repeat of what we saw earlier, if we look at the cross section of the tree, you've got the outer bark, the phloem, the cambium or secondary meristem, the sapwood, which is the living xylem and the tree is still using to move water and nutrients, and then the heartwood, which is the dead xylem, which is no longer moving water and nutrients. And you can see that in this stump cross section. The one thing that's different here is this is a white oak. It was cut about 6 months before I took this picture. You can see the heartwood here is still kind of a cream color. You can see a little bit of discoloration that's resulting from wood decay fungi getting after it, but the sapwood is completely black because remember the sapwood relies on active defense as opposed to the passive or static defenses of the heartwood. And because the sapwood was killed suddenly, there is nothing there to defend it. So this discoloration are the wood decay fungi that are beginning to break down the sapwood and consume it. And then here's the bark of the tree. In addition to what we've talked about, there are some messy chemical bits associated with the growth of trees. Auxins, which are produced in the buds, are hormones that can control growth form. The terminal buds will use, or apical buds will use auxins to suppress lateral buds lower in the tree. When the trees that maintain this have X current form with a strong central leader, and then there are other trees where the dominants lapse, and they have more of a decurrent form where they have some really large branches low down. Trees also have ways of sensing gravity, light, water, and other environmental cues. There are chemicals, and changes in the balance of those chemicals can cause the tree to either grow towards the gravity or the light or the water, or away from the sources of those stimulants. Trees also can sense their carbon and nitrogen ratio. Carbon comes from the canopy in the form of photosynthesis, nitrogen from the root system in the form of, well, of nitrogen. And depending on what that ratio is, it will direct energy to grow roots or to grow shoots. So if the carbon nitrogen ratio is high, that means it's got a lot of sugar, but it doesn't have the nutrients to do work with that sugar. The tree will grow more roots in order to raise or lower that carbon nitrogen ratio. On the other hand, if the carbon nitrogen ratio is very low because it's got lots of nitrogen and other nutrients that can be converted into tissues, but it doesn't have enough carbon to do the work, the tree will grow shoots. This is why you do not give newly planted trees nitrogen. In fact, you should not fertilize a newly planted tree for at least a year, because you want that tree to grow roots, because generally the trees we get from the nursery are not what's the word I'm trying to think of? They've got too much shoot, too much canopy, and not enough root system. They're out of balance. And there is another thing called phenotypic plasticity that can control how plants grow. And this is about how plants turn on or turn off genes. Some plants have more phenotypic plasticity than others, and as a result, they are more responsive to their environment. So, for example, oaks have a great deal of phenotypic plasticity, and the form they take depends a lot on the environment they grow in. So if they get lots of sunlight, they tend not to be as tall, and they put a lot of energy into growing these great big branches. On the other hand, eastern white pine does not have a lot of phenotypic plasticity. It grows the same way in the open as it does in the forest. Okay, I'm coming up on a place where I can stop and answer questions, but now, everything I've said so far pretty much applies to most trees. There are some exceptions to this. One exception is that monocots have no secondary meristem. They don't get bigger around as they get older. They stay the same diameter. In fact, the monocots, primarily the palms, only have one big chunk of meristem at the top of the tree that grows new wood, new leaves, and flowers and fruit. Conifers have no latent or suppressed buds in the crown, so they do not produce sprouts. Dicots, on the other hand, can produce sprouts when suppressed lateral buds are released, which are scattered throughout the tree. And the reason I call it Plan B is it allows for the replacement of lost parts of the plant. If the top blows out of a palm tree, it's dead. If a conifer loses all its branches on one side of the tree, it can't recover from that unless branches from the other side of the tree grow around. Dicots, on the other hand, can replace those lost parts. And so the reason this is important for you to understand is this is what leads to sprout formation. And there is a lot of misinformation about sprouts. People call them suckers. I've heard them say, you know, they're stealing water and energy from the tree. They're weakening the tree. And this is all bullshit. Sprouts form as an evolved response to stimuli. When they first start growing, yeah, they're a carb sink, but so is the new regular growth you get in the springtime. Once they get their leaves out, they become self-sufficient. They are not taking resources away from the tree. They are forming in response to pests or disease or wounding. So frequently, seeing sprouting on a tree is a bad thing because something else is going on. For example, this picture is an ash tree, and all the sprouting down here off the trunk is because this tree is being killed by emerald ash borer, and it's trying to replace that lost photosynthetic area with new sprouts down there. Sometimes those sprouts, though, are just occurring because the tree is accessing new resources. Say it's a new subdivision and they've cleared off all of the trees, and now a tree that had been in the interior of the forest is now at the edge, and it's suddenly getting a lot of sunlight. It's going to grow sprouts to take advantage of that. There are other types of trees, particularly around here, pawpaws and are particularly known for this, and sumacs. This is part of their reproduction. They just send up sprouts from their root system, but it is part of the evolution of the tree. They are natural. They are not in and of themselves bad. What's bad is what's causing the tree to put out those sprouts. And it's why I call this Plan B, but it only occurs in dicots. All right. So we got a question in the chat. Is xylem the same as sapwood? Hopefully, I covered that, but sapwood is a type of xylem, but heartwood is also a type of xylem. So xylem is wood. Sapwood is the part that is still biologically active in the tree and is moving water and nutrients. Heartwood is no longer active. Okay. I saw somebody had put their hand up.
Thank you. That was me. It's just easier for me to talk. I was typing.
That's okay.
So you had mentioned that, you know, when you get a tree from the nursery, you don't fertilize it, you know, plant it, right?
Right.
So earlier this summer in June, I had to transplant a camellia, and I had to.
You shouldn't fertilize a transplant either, usually.
So what about, does that include like soil conditioner and stuff like that for the clay soil?
Yes, that's a completely different discussion. Hopefully when you talk about planting, they'll talk about what something is called
because they died. I must have killed it. I don't know if it was the weather or I killed it, but.
Well, yeah, this past June was not a great time to transplant stuff.
I know it was
because we were going into some really rough weather then.
It was either that or a new subdivision just mowed it down, and it was an old camellia.
Well, you gave it a shot.
so
I mean, better you dig it up and try to transplant it than you leave it for the bulldozer. But yeah,
so don't throw the soil conditioner.
You probably left a lot of the root system behind when you dug it up, which is why you didn't want to fertilize it.
Okay.
But when you amend the soils, you create a hydrological discontinuity.
Mm.
Water does not like to move from one soil type to another soil type.
Ah, okay.
And so when you amend the soil in the planting hole, you essentially turn that planting hole into a pot,
Ah,
because water won't come from the outside of the planting hole into the planting hole until the outside is saturated. And similarly, and this is where people really get themselves in trouble when they start trying to improve drainage by putting sand or gravel or something in the bottom of the planting hole. The water is not going to move out of the planting hole until that amended soil is saturated. So the best thing to do is you plant the plant, you put your unamended backfill in, you do not give it any fertilizer, you mulch around the outside, and then you water it at least twice a week until it gets established,
okay.
which is 1 year per inch caliper, which is a good reason to plant small.
Yeah. Thank you so
So,
much, sir. I
You're
appreciate that.
welcome.
Hey, Jim,
Yes.
there was another question just now from Joanna.
Okay.
So should you never prune the small branches that start growing at the bottom part of the tree? I know you're going to get into pruning later, so.
I am. And I'm not going to say you should never prune them. You just should be aware of why they are there and why what's going on. Why is this tree producing small branches at the bottom? In some cases, like crab apples and a lot of the malus and prunus, that's just the way they grow, and you can leave them go or not. And especially I see in these ornamental cherries, they will put out what people call water sprouts, where they have these small sprouts that start growing vertically from the scaffold branches. And it's usually because of the stress of old age that they're doing that. So we'll talk more about pruning, but you do have to be aware or think about, why is this plant producing these sprouts? What's going on, and what are the ramifications of removing them? So there is a lot of literature out there. I've talked with master gardeners. I've talked with extension agents who, they're on a rampage about sprouts. And in fact, in the current version of the TreeStewards manual, they pretty much are telling you should always cut the sprouts off. And that's not the case. You got to figure out why it's sprouting. Okay, let's talk real quick about root growth, because it's pretty much the same thing as shoot growth. You got the root and the primary meristem at the bottom, and in this case, it makes new cells, and then they elongate and differentiate into all of this stuff that allows them to collect water and nutrients and move them back in, move those resources back into the rest of the plant. So here is a 1-year-old red maple, which was grown from seed. This came from the DOF nursery. You can see the tree, this little tree produced a lot of roots. So these really fine roots, and these are actually not the absorbing roots that you can see. You really can't see those fine absorbing roots. And then the connective roots, and up here is where you would have the heart roots and sinker roots, which are part of the buttress system. The other thing to keep in mind as we go forward is that the tree is constantly growing and shedding those absorbing roots, because what's happening is it senses, oh, there's a water pocket over there, and it grows a root over, but once it drains the water pocket, it abandons the root. And for the most part, what we're looking at is there's almost the same volume of shed absorbing root mass as there is the leaf fall. that occurs if it's a deciduous tree. So it's doing it a lot. All right, woody plant sex. So I'm going to go through this. They go into really way too much detail on this in the manual. This is an angiosperm, and what makes it an angiosperm is this thing, because remember, angiosperms mean covered seeds. This structure is called the carpel. And within the carpel are the ovules. And then the style and the stigma, which I just recently learned, if you ever shuck corn silk is the style and stigma that's attached to the carpel, which contains the ovules. And once those ovules get fertilized, the style and stigma comes off and becomes the corn silk. But this is what makes it why they're called covered seeds, because what will happen is after the ovule, the ova in the ovules are fertilized and they start producing viable seed, the carpel will start to expand and create the fruit that goes around it. And the carpel has three sides. There are three parts. There's the endocarp, which is the inside, the mesocarp, and the exocarp. So the best way that I know of to illustrate these is if you cut through an apple, not from down from the stem down to the bottom, but across, what you will see is there are 5 little chambers that have this really hard kind of, it's almost like fingernail quality lining. That hard stuff is the endocarp, which is on the inside. Then all of the flesh that we use for jelly and jam and pies and whatever, that is the mesocarp. And then the endocarp is the skin of the apple. So in a what we call a perfect flower or co-sexual flower, you have the female part, which is the pistil with the carpel and the style and the stigma. And you also have the male part, which is the stamen, which has the anther and the filament and produces the pollen. But you can also have, in the angiosperms anyway, female flowers that do not have anthers and male flowers that do not have pistils. And what we see in trees is that they get, you can find almost any combination of male, female, co-sexual on any tree, on trees. So it's very common for trees to be monoecious. That means they either have co-sexual flowers or they have male flowers and female flowers on the same plant. So, for example, oak are monoecious, but they have male flowers, which are those catkins that fall off and make a mess in the spring, and female flowers that become acorns. Then there are dioecious plants. The poster child for this is holly, where you have male flowers on one plant and female flowers on a different plant. Oh yes, the other point to make about monoecious is they could be co-sexual flowers. So tulip poplar has co-sexual flowers that have both male and female plant parts in the same flower on the same plant. Then you run into some other combinations. For example, polygamodioecious is co-sexual male or female flowers on the plant. So it's basically a dioecious plant, like the holly, that only has male or female plants. But the polygamo means that they'll have some co-sexual flowers on every plant. So, for example, Nyssa sylvatica is either mainly male or mainly female, but it always has some co-sexual flowers on both of them. Then there's polygamo monoecious, which can have co-sexual male and female flowers on the same plant. And the maples are probably the most adventurous because you can have maple trees that are all male flowers, which means you don't have to worry about helicopters, or all female flowers, or male and co-sexual, or female and co-sexual, or they can have a male branch and a female branch on the same tree. So the maples are taking advantage of all possible combinations of these things. My neighbors, before they butchered it, had a silver maple that looked like a barber pole because in the spring when it was producing the flower, the Samara, the helicopters, it was kind of striped on the tree. It wasn't the whole tree, but because it had some male branches and some female branches. The Wikipedia entry on plant reproductive morphology that has all of these names in it has about 20 different terms for the way flowers are arranged on angiosperms. Keep in mind that gymnosperms do not have flowers. They have cones. The other thing that you can run into with tree sex is self-incompatibility. It is not a good idea to date yourself. And there are a number of different ways that trees do this. One is through dioecy. This is, the separate male and female flowers on different plants. Then there is herkogamy, where the flower structure is such that it separates the stigma and the anthers, so they can't get together and pollinate them. Dichogamy is the pollen and ova mature at different times. This is what happens on pawpaws. And then there are a whole bunch of other things that go on from the pollen won't germinate, the pollen tube won't grow, because when pollen hits the stigma and the pollen germinates, it grows a tube down the style so the male gametes can get down to the ovule. In some cases, the ovule will refuse pollen from the same plant or a genetically close plant. And in other times, it just won't develop an embryo. Chestnuts are self-incompatible, but they still produce the burrs. It's just that when you open up the burrs, there's this whizzing, it's not really a nut inside. So here are a couple of examples of herkogamy. This is in Rhododendron atlanticum. This is one of our native, actually not native to here, it's a southeastern Virginia native deciduous azalea. And you can see here are the anthers and here are the stigmas, and there's no way this anther is going to touch that stigma. So it has to be pollinated. And then penstemon, which is not a woody plant, but it has its anthers up here and its stigma down here. And there are some bees that have that specialize on penstemon that have pollen collection structures on their backs so that they can interact with these anthers up here. All right. We're going to talk about water and gas exchange. This is caused by, in most cases, by partial pressure differentials. And what that means is essentially there is a concentration flow that happens. So if you have two areas that have particles that look like this, and you open a channel between them, what will happen is some of these red crosses will move over here and some of the blue stars will move this way. And it's not so that you have the same number of red crosses and blue stars on each side. It's so that here the partial pressure of blue stars is 75%. Here it's 75% for the red crosses. And once they move, it's 50% for each of them. And so the idea is you want the same number or the same percentage of that molecule in the volume on both sides. If you had a semi-permeable membrane, so only the blue stars could move, what would happen is the blue stars would move over until the partial pressure of blue stars in this volume is the same as it is here. And in this case, it's 50% and 50%. And the red crosses do not move because the opening won't allow that. So how does this work in terms of the plants? Well, this is a micrograph of the bottom of a white oak leaf. It shows the stomata there. All those little dots are stomata. And what happens is you have leaf gas and atmospheric gas, and the plant through photosynthesis is pulling carbon dioxide out of the leaf gas and pushing molecular oxygen and water vapor into the leaf gas. So when those stomata open, the partial pressure of carbon dioxide in the atmosphere is higher than it is inside the leaf. So carbon dioxide goes in, but the partial pressure of oxygen and water vapor on the inside of the leaf is higher than on the outside. So those two gases go out. Lenticels work the same way, but in reverse, in that it's oxygen going in and carbon dioxide coming out. So the stomata are associated with photosynthesis, and the lentils are supporting cellular respiration. The same thing is going on in the soil, where you have a greater concentration of water molecules outside of the root than you have inside the root, and the plant can actually control this by pushing nutrients or other molecules into the cells in the root system, and this creates osmotic pressure that pushes water into the root system. It also works in the mycorrhizal fungi are collecting water and nutrients this way. One of the things we didn't talk about is nitrogen-fixing bacteria that can live in the roots of some plants. You may have heard about that for legumes, but it also works for spruces. And so when you look at this picture, you can see all those little nodules where the nitrogen-fixing bacteria are living. So this process of molecules moving from areas of greater concentration to lesser concentration is what brings water and oxygen and carbon dioxide into the plant, and also moves water vapor and oxygen and carbon dioxide out of the plant. Now, once the water gets into the plant, you got to get it from the root to the crown. So that partial pressure differential is causing water vapor to move out of the leaves through a process called transpiration, and that can cause suction on the vascular tissue that helps pull water up from the roots. But the physicists are fond of pointing out that suction pumps only work up to 32 feet. So something else has to be going on to get water from the root system to the top of really tall trees like redwoods without losing pressure. Part of that is osmotic pressure that is helping push water up from the roots. But there are other things that are going on. For one thing, cellulose has an affinity for water. So and you can do this after class today. Get a piece of paper towel or toilet tissue and put a corner of it in water, and you will see the water climb up through the cellulose because of that affinity. And that may be part of what's helping move water up the column. The bottom line is we know that transpiration suction is part of it. Osmotic pressure is part of it, but we don't really have a definitive answer on how water gets transported from the roots to the crown. Okay, now there is one other thing that trees need to absorb in order to support their life functions, and that's nutrients. Nutrients are not taken up through these partial pressure differentials. In fact, the way that plants get nutrients into themselves works exactly opposite of the partial pressure differentials. So you can see in this picture, we have a lower concentration of nutrient ions outside of the root than we have inside the root. Normally, that would cause nutrient cations to move out of the root and into the soil. But the plant uses active transport to get the flow of nutrient concentrations to move the other way from low concentration to high concentration. So what's happening here is all of our nutrient cations are water soluble. As osmosis is pulling water out of the root, out of the soil into the plant, it's also moving water towards there. And then the plant can use active transport to bring in the nutrients that it needs. And this is usually done through some kind of a molecular channel in the cell membrane. There's a whole lot of very complicated biology and chemistry that's going on here that we're not going to get into, but that's how nutrients get in. Everything else goes in through this physical process that moves molecules from low concentration to high, or from high concentration to low concentration. But the nutrients are different. Any questions? All right. One of the things.
I just said I don't see any.
Okay. So the second part of what we're going to do tonight is talking about pruning. And in order to talk about pruning, you got to know how branches are attached to trees. So as trees grow, remember the secondary meristem kicks in and it creates secondary growth in the branches and in the trunk. And in general, this moves from the top or the tips of the canopy down into the trunk. So the branch will get a new layer, and then the trunk will grow a new layer of wood, and then the trunk grows a new layer of wood over it. And when you have good branch attachment, what happens is the fibers and the wood from the tree or from the trunk and the branch kind of wrap each other and weave together. And a good branch attachment is going to reach into the center of the tree. It's going to be U-shaped, and it's going to have what's called a bark branch ridge. And you can see this little kind of dimple here is an indication that this was a good branch attachment. So this is how Alex Shigo drew it up, and it's still pretty much the same. You can see the branch gets bigger around, then the trunk wraps over it. And there's your bark branch ridge. This is what it looks like on the interior of the tree. Unfortunately, this was a fairly large branch on my willow oak that failed, but you can see how the wood growth wrapped in through here. When it comes to pruning, you got to know whether you have a good branch union or a bad branch union. Good branch union, there is that branch bark ridge. And what that means then is the wood underneath here is being wrapped and woven together, whereas this is a bad branch bark ridge. There is, or a bad union, there is no branch bark ridge. There is included bark here, and you can even see the seam that travels a long way down the branch. This is why you don't want to plant calorie pears, especially the Bradford cultivar, because this is what all the branch unions look like. And this is a Bradford cultivar, and this creates a weak spot in the tree. Another thing that's important to understand is how trees respond to wounds. And this is a process called coating. So remember, only the meristem can make new cells. So if the tree gets wounded, if it gets a hollow space in it, that stays with the tree forever. It never replaces those lost tissues. What it will do, especially if that that cavity is created by a wood decay fungus or wood decay organism, is the tree will try and seal that off and prevent it from spreading in the tree. Now, one of the things to understand is there are two types of fungi that will attack a tree. There is a wood decay fungi, and this is the same kind of a fungi that causes dead trees lying on the ground to decompose, or if you leave, say you've had a fireplace and you've got a stack of firewood that you're not doing anything, don't do anything with, eventually that will decay and go away. That is a wood decay fungi. There are also pathogenic fungi, and these are the ones that attack the sapwood and kill and consume living cells. And although the tree uses the same principles, it's a little bit different if it's in the heartwood where you have no living cells and you're dealing with a wood decay fungi, and whether it's happening in the sapwood where you do have living cells and you're dealing with a pathogenic fungi. The other thing is those wood decay fungi do not kill the tree. They can weaken the tree structurally by creating cavities and reducing the amount of sound wood in the trunk of the tree, but they can't kill the tree because they're not affecting the biologically active cells in the tree, whereas those pathogenic fungi can kill the tree because they are killing the biologically active cells in the tree. So this process of coating creates barriers to prevent either wood decay fungi or pathogenic fungi from spreading in the tree. And some of these barriers get laid down during growth. Others are created in the process of making heartwood, and some of them only occur after there is an attack. So these compartments are made of four types of walls. The weakest wall is wall one. This is where the vascular tissue gets clogged up to try and prevent these organisms from moving up and down in the tree. Here it says wall one is incomplete until there's an attack in the sapwood. However, the process of converting sapwood to heartwood creates, part of that is to create that wall one, because there's nothing in there in the heartwood. There are no living cells to clog up the vascular tissue. Wall two is the second weakest wall, or second strongest wall. Wall one is the weakest. Wall two is next. And this is laid down during growth. So at the end of the growing season, the cambium will lay down a relatively thick wall of parenchyma at the edge of the growth ring. And that is there, of course, the idea is to keep the pathogen or the decay fungus from moving deeper into the tree, towards the middle. The side walls, in die cuts anyway, are those parenchyma rays that I showed you earlier. Those are also laid down at the time the tree are during growth of the tree. And of those walls, it's the strongest. In sapwood, it's full of cells that can produce defensive chemicals. In the heartwood, it is also full of cells, but they're not producing defensive chemicals, but they have imbued themselves with defensive chemicals. The fourth wall, which you don't see in this diagram, is the new growth that occurs to seal off the wound. And so it would look something like this. As the tree is starting to seal the wound, the cambium will form what is called callus tissue as part of the cambium, and that creates wound wood that has this curved look to it. And eventually what will happen is that wound wood will come together and it'll graft together, and you wind up with this entire circle here. The one thing that's wrong with this picture is it doesn't show the decay that's still there, or the pocket, the cavity that is still there from this decay, because you're not going to build new tissues around it. So in terms of the micrograph, there is wall one, which is those vascular tissues that are clogged up either by the parenchyma or the existing parenchyma if it's sapwood, or it's clogged as part of making the heartwood. Wall two are these radial parenchyma, and although it usually is talked about as at the end of the year or at the end of the growing season, you can see that during the growing season, they're also laying down these lines of parenchyma that will help prevent inward growth of the decay. And then wall three are those parenchymal rays, and then wall four is the new growth that goes around it. And you can see from this picture, this is what I mean by that new growth. There is a cavity here. If this branch had long enough, eventually what would happen is these ram's horns, is what we call these little things, would have grafted together, and you would have solid wood growing over it, but you would still have this kind of T-shaped decay process. And this is further down in the trunk of the tree. So this is a closed cavity. This is an open cavity. And you can see the process here. You've got this decay cavity here. You've got those ram's horns, that wound wood that is trying to come together and graft it, but you can see the seam that runs up through here. So there's a pocket of, or a cavity that runs all the way through that tree.
Hey,
Another thing I want to talk, yeah.
I'm sorry to interrupt. I was just wondering, before you go to the next topic, can you tell what CODIT stands for?
Compartmentalization of decay in trees.
Yeah, thank you.
Okay. So tree growth and hormones, and I'm not going to get real deep into this, but remember I talked about how as the tree grows, it puts down, or at least as dicots grow, it produces these lateral buds, which are suppressed by the terminal bud, the hormones produced by the terminal bud of the tree or of the branch. And you can see here all these little pimples, those are those suppressed buds. When the buds are released, we get sprouts that looks like this. And as I mentioned, if the dominance of that terminal bud or apical bud is continues throughout the life of the tree, you'll get a structure that looks like this. So this is a lot of the pines look like this, but this is also what you see in, for example, sweet gum or tulip poplar. They both are excurrent trees that have a very well-defined trunk and relatively small branches coming off. Almost all trees start off their life this way, but eventually they lose that apical dominance and they can start becoming decurrent. And when that the timing of that depends a lot on where the trees are growing. For example, with the oaks, they may not become decurrent and lose that apical dominance until the tree is 50 or 60 feet tall. And then you've got your high, relatively thin crown up there, and then this long, straight trunk that doesn't have any branches on it. That's what happens in the forest. Out in the open, though, it loses that dominance and it starts producing, it gets that decurrent form and starts producing lots of branches. So how does all of this relate to pruning? Well, before we get into pruning, you have to remember, when you prune live tissue from a tree, you are wounding that tree. You're reducing its ability to make food. You are potentially opening pathways for pathogen. And so what you are doing when you prune live tissue is you are forcing the tree to expend energy on wound closure and fighting pathogens. And at the same time, you're reducing the tree's ability to produce food. So you need to be thinking about that trade-off before you start making pruning cuts. And this gets back to that question about the sprouts. Why is it producing sprouts? How badly does the tree need those sprouts? If you think the tree really needs the energy from those sprouts, leave them be. If not, you may be able to prune them off. And so this is what it means to prune. You start by observing the plant. What do you see going on with the plant? What's wrong with it? What are things that maybe need to be corrected? Your observation may be it doesn't need anything, and then you're done. Once you've observed the plant, you set your objectives. What do we want that plant to look like? Then you make a plan for how you're going to get there, and then you make your first cut. And after you make that first cut and you get that branch out of the way, you go through the whole process again, because you may visualize something in the first part of the process that doesn't turn out to be what happens when you make your cut. So there are a bunch of reasons that you might prune. The most defensible reason is to maintain the health of the plant. It may have some kind of a fungal disease out in the edges of the canopy, and one of the ways you deal with that is you prune out those affected branches. You also may prune to reduce risk of failure, because remember the included bark and the bad branch union, that is something that could potentially compromise the whole tree. Another reason we prune is to provide clearance for pedestrians and vehicles. Sometimes we prune to associated with flower and fruit production. Sometimes people prune trees to reduce shade, and my response to that is grow more shade tolerant plants under your trees, and then to improve the view or the aesthetics of your property, but I don't know how you can improve by reducing the amount of tree. Nothing is prettier than a tree. And I meant to get rid of this. So in the natural pruning system, which you're going to learn about more about on Saturday, there really are only two types of cuts. One is the removal cut. So this is working with that coated process to remove a branch. And trees naturally lose branches in the forest because the branches have to provide all of the energy they need. And as they start to get shaded, they may not be able to produce enough. The branch dies and the tree needs to get rid of it. So they've evolved methods for doing that. And in particular, they have evolved what's called a branch protection zone. This is the biology inside of it, and it's visible as the branch collar. And so at the base of the branch, you'll see this swelling where those fibers are being woven together. And what's happening down there is the branches stored a lot of defensive chemicals to prevent decay from getting in through the wound where the branch died. And the cambium is primed to create this wound wood that closes the wound. And this, by the way, is why you prune off, one of the reasons you prune dead wood off of the tree, but you want to be sure you leave that branch collar so that it can close that wound as quickly as possible. In the book, I think they have this, I think this is page 63 currently, the removal cut. And essentially what you're doing is you cut, and the science on this has changed a little bit from the earlier editions of the book, of the manual. But what you want to do is start outside the branch bark ridge and cut parallel to the axis of growth and remove that branch. That'll preserve the branch protection zone and allow it to the tree to heal, or at least work with it. This is the old method where you visualize a line that runs perpendicular to the axis of growth, and then you bisect the line to make the cut. Unfortunately, this method leaves us with a lot of cuts that look like this, and you can see where the tree is trying to seal this off, and it's got this little nubbin in its way. That's why we now are saying you should just start here and cut straight down, and that way you don't include part of the dead branch in your cut. Okay. Now, sometimes you have to do a removal cut where you have included bark, and this is from last year's advanced training. So this is an elm over there by Arlington Community Center, and you can see this is a bad branching in, which is not that uncommon with elms, and you can see the included bark here. And when you've got this situation, what you do, and somebody I think may have started the cut or lined it out, you cut from the outside in towards the included bark, and you wind up with a wound that looks like this. Unfortunately, I mean, this is not a great outcome, but it's all we can, or the best we can hope for, because otherwise this branch will get really big and it'll tear off the side of this tree and make a big wound on it. This cut actually could have been done a little bit lower to reach in. And the tricky part here is you want to cut all the way through the piece you're removing, but you don't want to cut into the included bark here. And I know it looks bad because you've got this relatively large wound, but keep in mind, this is bark. This part is already covered. And it's always good to make this cut maybe a little higher than you think it should be, because you can go back and make another cut underneath to reduce the size, the cross section of this wound. But you can't put that back on once you've cut it off. The other thing is you want it to slope so when it rains, the water doesn't occur there. And somebody had their hand up.
I don't see a hand up.
You
Okay.
answered my question as you went along. Thank you, sir.
Oh, okay. The other type of cut you're going to make, and this one's a little more complicated, is called a reduction cut. This is shortening branches by working with hormones or not. So let's start with the way we would make a reduction cut. And so this is where, for some reason, you need to shorten the length of that branch. And I'll talk about, there can be a number of reasons. It's not just because, you know, the branch is growing up into a power line or there's something overhead. It could be that the branch is getting overlong and reaching too far out, so you're taking some of the weight off the end of that branch. But it can also be to control the growth of the branch so that you can safely do a reduction later on. And so this is why it's called, or a removal later on, this is why it's called a reduction cut. What you're doing is taking away the larger part at the branch union. So this part remains and this part goes away. And so you can see here in this picture, this dotted part is what's being removed. And you make the reduction cut in pretty much the same way you make the removal cut. You go from your branch bark ridge, you visualize a line, or you can draw a line that is perpendicular to the axis of growth. And then you bisect the line or the angle from the branch bark ridge and that visualized line. So you're cutting along here, and notice it's not straight down across. It's not flat across. And what this does then, by making this cut this way, is it allows the terminal bud of the lateral branch to take over, suppressing the buds below the cut. If you were to make it straight across like that, all of these buds over here would erupt into sprouts that come up. That's what happens when you see a top tree. You get lots of those things. So making the cut this way is trying to prevent sprouts. And here is an example. This is my red maple. The part that I cut off was growing down into the middle of my driveway. So I did a reduction cut in 2008. This was what it looked like a year later. This is what it looked like 3 years later. This is what it looked like 14 years later. Notice no sprouting along this branch because I made this cut in such a way that eventually, or that the terminal bud of this lateral could take over, suppressing those buds. Now, remember I said this is working with the hormones to prevent sprouts or not. Sometimes you want to do a rejuvenation pruning. So this is my Amorpha fruticosa. It's also called indigo bush. And it was getting kind of leggy and big. And we were sort of ambivalent about it. I wasn't sure that it would respond to this, but what I did here was I made reduction cuts, not at branch unions, but at buds. So there is a bud out here. And by doing that, it allowed the release of a lot of buds in the interior of this branch or tree. This actually is just 1 year's growth, but you can see it has really rejuvenated it. And these sprouts are there now to replace all of the lost photosynthesis synthetic area that we had here. Sometimes arborists will do this on old trees that are starting to grow into retrenchment and starting to have bald spots in their canopy, they'll go in and make these cuts, only they're doing it in a way that is causing sprouts to occur that will help fill in some of the spots in the canopy. There are other times when we are making other applications where you make cuts specifically to create sprouts that you can then manage. This is at an apple orchard, and you can see it has all these sprouts on it, and they pruned it this way so they could get those sprouts because they produce more flowers and they will produce more fruit, but it also keeps them from getting too big and competing with each other. In terms of the type of pruning you are likely to be doing, this is doing everything wrong, but they do it wrong on purpose because they're trying to produce those sprouts. Here is another example of managing the sprouts. This is called espalier. So this is where you're trying to grow a tree that essentially is two-dimensional. I don't know how well you can see it in this picture, but the tree is growing up this way, and it's got branches that reach out along this fence line, and they're training the tree to do this. This is actually at the greenhouse at Mount Vernon, because George Washington did this with his fruit trees so he could grow fruit in the same space that he was growing all of his table vegetables for his kitchen. Now, in your book, they talk about, they don't call it a watch out situation. They talk about the 1/3 rule. And they will tell you you should never remove a branch that is more than 1/3 the diameter of the trunk. And that you should only reduce to a lateral that is 1/3 or greater the diameter of the part you're removing. In the profession, and these used to be rules in the profession, and we have gotten rid of the rules, and I refer to them as watch out situations. So if you are thinking about doing a removal of a branch that is more than 1/3 the diameter of the trunk, you might want to think about it again and maybe think about possible alternatives. That's why I call it a watch out situation. There could be bad things that will happen. The same thing about reducing to a lateral that is less than 1/3 the diameter of the removed part. That could mean that that lateral is not going to be able to successfully contain the sprouts. But in some cases, you want sprouts. So you just have to be aware of what those potentials are. And that's why we call it why I call it a watch out situation. Any questions? Okay. There is something that you will hear about called the 3-cut method. This is what we use primarily for removals of larger branches. And by larger, I mean one that you cannot control with one hand. So if you can't control that branch with your one hand and arm, you should be using the 3-cut method. And the reason it exists is because of something called the shear plane. So here's the trunk of the tree, and here is a branch that is sticking out from the side. And because of gravity, that branch is being affected by two different forces. The lower part of the branch is under compression because gravity is pulling it down. At the same time, the upper part is under tension. And then where we transition from tension to compression is called the shear plane. And sometimes branches will crack like this. They'll get a crack, excuse me, running the length of the branch along that shear plane. But when we cut, we use this 3-cut method to prevent that from causing the branch from splitting and tearing off the trunk of the tree. So your first cut you make on the underside of the branch. And you go up about 1/3 to 1/2 of the way into the branch, usually until the saw starts to pinch. The second cut, you go further out the branch and you cut straight down through the branch. And then the final cut is where you make that cut, that removal cut that protects the branch collar of the branch. Because with this little stub, you can control that and you're not going to get the splitting. So here's what it looks like. You can see here kind of that I made the first cut on this pruning cut. Then as I started to cut down through the branch, it eventually got to a point where it split. But it only split to that first cut. And that's what that first cut is there for, is to prevent it from splitting and ripping all the way down the length of the branch and possibly into the branch collar and into the trunk of the tree. All right. One thing you need to know is that proper pruning cuts do not need to be sealed or dressed. This is particularly true of the stuff they sell in the nurseries as wound sealant or wound dressing. There are some times when, if you are pruning the tree and there are fungal spores flying, that you might treat that cut with a fungicide. This is not sealing it. What you're doing is you're putting a chemical in there that's going to kill those fungal spores. So it could be as simple as spraying it with Lysol or some other antifungal spray like that. But it's not the stuff they sell in the nurseries that they say, oh, you can also fix your pots and your sidewalk and your gutters and all kinds of other stuff with it. That just gets in the way of healing. So it looks like we got a few minutes, so I'll go through this example of how we apply some of this stuff. So this is my crabapple. You just saw a picture of my of the two-cut method to remove a branch. In October of 2008, I looked at the tree, evaluated it, did my observations, and what I observed was it had this one branch that was becoming a codominant leader. It also had another branch down here that had an aspect ratio, that is the ratio of the branch diameter to the trunk diameter of about 1. So this branch is almost as big around as the trunk of the tree, and it was growing out towards the street. Ideally, I didn't want this branch here because it was growing out towards the street, but I knew, because the aspect ratio is close to 1, which is well over 1/3, that if I were to just take my pruning or my loppers and cut that branch off in October of 2008, I would make a big wound on the trunk of this tree, and it might very well result in rot getting into the tree and killing the tree. The other thing was that this is a fairly young tree and it had a lot of growing to do, so I wanted to be careful about how much photosynthetic area I took off of this tree. Now, it being October was not a big deal for this growing season, but I wanted to be sure I had a branch there that was going to support leaves and help contribute to the energy of this tree over the coming growing season. So what I did was, instead of just pruning these two problematic branches off, I set my objectives, which was to not have a competing leader and not have a large, low, vigorously growing branch on there. And my plan was to do reduction cuts reduce both of these branches so I could slow down the growth relative to the trunk of the tree and potentially keep this, at least this branch, as a scaffold branch for the tree. This one was too low and growing in the wrong direction to be a good scaffold branch. So I knew I wanted to take that off, but I knew I wanted to wait until it would be safe to remove it. So for the codominant leader, I went out, I found a good lateral here, put my pruners up there to visualize that line and create the angle. And you can see I rotated my wrist a little bit before I made the cut, and it looked like that. And this is what happened. This is when I made that reduction cut. 3 years later, look at the difference in the size of that potential competing leader with the trunk of the tree. And what happened was, and then by 2016, you can see it's even smaller. So what happened was, by making that one reduction cut in 2008, I turned this branch from a potential competing leader and a problem into a scaffold branch, which is still on the tree today. You can see what happened to the cut. This is what it looked like right after I made the cut. This is what it looked like 3 years later. Notice that this whole thing is kind of evened out. So it really doesn't look like at one point this was a side branch that was smaller than this one. And that cut has completely healed over and I can't find on the tree anymore. For that low vigorous branch, I did a reduction cut here in 2008. This is what it looked like in 2008. 3 years later, notice the difference in the aspect ratio. Well below 1/3. So now I can cut this branch off, and I did in just 3 years later. But notice it took 3 years to go from making my observation, setting my objectives, making a plan, making a cut, making another observation 3 years later, setting the objective, making a plan, and then making the cut, and finally removing this branch from the tree. Okay, we're a little over, but I do want to talk about this. Root pruning. As I said earlier, trees do, well, excuse me, let me back up. Trees are not as well evolved to lose roots because of they're in the ground, so they don't have to produce their own energy. You've got this dendritic pattern of growth, so the larger the root is, the more the root system is connected to it. Those absorbing roots, however, are being shed and regrown constantly. And there is a rule of thumb for root pruning, which is essentially that if the root is smaller than your thumb, or for you ladies with small hands, maybe smaller than your husband's thumb, a root that's about 1 inch in diameter, they sprout, they regrow and grow rapidly, and they will also seal rapidly. So you can cut those roots that are under 1 inch in diameter pretty much with impunity. One thing to be aware of is soil will dull your saw or your pruners fairly quickly. A common problem that people have is they buy a plant and it's really pot bound, and it's got roots that are growing in wrong directions because they've reached the edge of the container and they started turning and growing weird places. And this is going to be a problem later on as the plant grows. You want to get rid of these roots. And the way you do it is you prune the root ball. In this case, I took my bow saw with an old blade and I sliced off about a 1 inch cord. A cord would be 1 inch, 1 inch from where the cut was made to the furthest outside edge of the root ball. And I went all the way around it. And I looked at the bottom, and it turned out I didn't need to make a cut, but sometimes you have to cut the bottom off as well. And so you reduce the size of the root ball. And I know this looks scary because you're cutting off all these roots, but remember what I said. These are the kind of roots that the tree is, or the plant is constantly growing and shedding. But by doing that, I have cut through this root, which is significantly less than 1 inch in diameter, at a point where it is growing in the right direction. So when it starts to regrow, it's going to continue growing in the right direction. One thing I will warn you about that doesn't have to do with pruning, though, is if you have a root ball like this, these things are sponges. They are going to suck the water out of the ground, out of the root ball very rapidly, and not necessarily until and it won't be until they get really established and start having roots growing out into the surrounding soils that they're going to be pulling water in from the surrounding soil. So you do need to water these frequently. This is Clethra alnifolia hummingbird, which is a gibberellin challenged Clethra alnifolia. And it's growing right by we planted it near the front door, so we were able to keep an eye on it and make sure and remember to water it. Quickly talking about pruning tools, your basic tools are going to be your pruning shears and your handsaw. Pruning shears should be bypass, not anvil. If you buy a folding saw, make sure it's got a good lock on it, because you do not want this thing to fold on your hand while you're using it. You may also want to have some loppers, which are similar to the pruners, just they have bigger jaws and longer handles, so you've got a lot more leverage on them. Some people will use a bow saw. One of the advantages of the bow saw is that it's got a longer blade, so you're getting more cut every time you go through it. But in general, that handsaw should be enough for anything that you do. If you want to go with something where you're pruning above ground, well above ground, you might get a pole saw or a shear. In this case, it's a bypass lopper here and a regular saw blade here. These things are kind of hard to work with, so you might want to try one before you buy it. And this is the, as I said, bypass shears or loppers, not the anvil shears, because what happens when you cut with these anvil shears is you'll have one blade cutting down to this flat plate and you're crushing the tissue rather than cutting it. If you have hedges or, well, pretty much hedges, you could get some hedge clippers like this. These, again, are bypass. You want to take care of your tools by keeping them clean and sharp. The blades can be sharpened with a stone or a file. Saw blades, you can sharpen those pruner saws if you have the right kind of file. You may need to take it to a professional to get them sharpened, or you can replace them. That's the other advantage of the bow saw, is a replacement blade runs about, well, the last time I bought one, it was $5, and the hand saws were, replacement blades were almost as expensive as getting a new saw. You should clean your tools after every use to remove any plant residues, any sap or anything stuck to it, and then wipe it down with an oil cloth or spray it with some WD-40 or whatever, to keep the water from getting at it and keep it from rusting. If you think you may have been working with an infected plant, you should disinfect. I use a 10% bleach solution. Somebody pointed out that that can lead to corrosion of the blade. So it's perfectly fine if you've got a lot of residue to wash the blade, disinfect it, wash it again, and then dry it off and wipe it down with the oil cloth. Remember to also keep your pivot points oiled. If you happen to have some heirloom tools, keep those handles painted or varnished. And when you are using your pruners, you don't want the branch, you want to get the branches close to the pivot point. And if it won't fit into the jaws, that's too big a plant to use that tool on. And again, if you are working with a diseased plant, you should disinfect the blades between each cut rather than waiting until the end of the thing, because you can be moving the disease from one part of the plant to another part with your blades. Okay. That's it. I was not too far over. Do you have any questions for me? I see one in the chat. One thank you in the chat. You're welcome, Kristen. Any other questions?
I don't know. Lori, did you have any?
No, but there are no more questions. Then thank you so much, Jim.
You're welcome.
Really, really appreciate it. It was such a great talk as always.
Okay.
All right, buddy, let it percolate.
You
And
guys have fun on Saturday.
Thank you.
indeed.
You're welcome.
Thanks
Good
so
night.
much. Bye. Bye,
Bye-bye.
everybody.
Bye.
Bye.